Para mantener la velocidad yo voy a hacer mi charla en inglés, pero las preguntas yo puedo responder en español. I want to talk about microalgae for production of biofuels, food and chemicals, and give a wider perspective of the technology. The content of the talk is I will briefly introduce the topic of biodiesel. It has been introduced already, but I will briefly summarize it. We started our research in this field with a feasibility study, an economical feasibility study, and I will show you some results of that. The importance of biorafinery of microalgae, so to make more than one product, more than just biodiesel from microalgae, and then I will cover a few of our research projects. Biodiesel, it has been mentioned before. There are, I think, two interesting ways to produce biodiesel from microalgae, and one also mentioned in the presentation of Emilia is Botryococcus. It actually does not produce lipids, but it produces alkanes, and those alkanes can be present in very high concentrations. Up to 70% has been observed, and the advantage as well is that actually these alkanes are more or less spontaneously released from Botryococcus. So Botryococcus is in that respect a very interesting microalgae. Nevertheless, it is not the organism where we generally talk about. If we talk about biodiesel production from microalgae, then we talk about lipids, and preferably about the acylglycerides. Algae contain like 20 to 60% of lipids, most of them 20, some of them 60. And they are considered to have a high productivity. If you compare it with palm oil, also mentioned in the first talk, about 6,000 liters per hectare per year can be produced by palm oil, and microalgae can produce a lot more. It is estimated something like 20,000 to 80,000 liters per hectare per year should be possible. 20,000 maybe today, 80,000 will need some time of development. Another advantage is, but nevertheless, 20,000 liters per hectare per year is considerably more than in other crops. There is no competition with food, and the main cause of that is that you can grow algae on salt water if you do it properly and if you do it with the right species. So that is the reason that microalgae was so much in fashion. We did a couple of years ago a feasibility study, and it was financed by Delta, and Delta is one of the electricity companies in the Netherlands, and it was done by Maria Barbosa. What we did is to have an idea about the cost of production of microalgae. We made on paper designs of plants to produce microalgae, and we did it at different scales. We did it at the scale of 1 hectare, and we did it at the scale of 100 hectares. We used 3 different systems, and the choice of the systems was based on the fact that we could realize that today. There are many new technologies, but those technologies are not ready to be scaled up to that scale, so it is difficult to do the calculations. So we used horizontal tubes, we used an open raceway pond, and we used flat panel reactors, and we have seen passing by those reactors already a few times today. And we made a very conservative calculation of the cost. So not only the design of the reactor, but all the maintenance, the tubes, the pumping, we have to buy the salts to produce the algae. We have to use CO2 to buy the CO2 to produce the algae, and we even decided to do it in the Netherlands because Delta is a Dutch-based company. And the reason that we did this was to see what the costs were and to see what were the individual cost factors in order to judge whether there is a possibility to really reduce those cost prices. And this is just one example. It is the example of the tubular reactor. And if you design such a reactor on a scale of 1 hectare, we get to a cost price of biomass, and that's per kilogram of dry biomass, of about 10 euros per kilogram. Much too expensive for biodiesel production. If you imagine that half of it is lipids, and then you have to convert it still to diesel, then you get a diesel price of more than 20 euros per liter. It's a bit too much. If you scale it up to a scale of 100 hectare, you can be much more efficient in your labor, especially in your labor. There are other things where you get more efficient in, and that results in a reduction of the cost price to something like 4 euros per kilogram of biomass. And these results are more or less the same for all the three systems. An important contributor in the system is energy, just energy to pump water around. Now, why is that so high? We have to realize that we based our design on existing technology. The existing technology is usually used to make high-value products. Then the biomass has a value of, let's say, 100 euros per kilogram. And if you have then a cost price for your energy of about 2 euros per kilogram, that is not really an important factor anymore for the company who is developing it. So many of those plants that are there have never been optimized in an energetic way. And if you go for a biodiesel production, you would have to do that. But nevertheless, we got something like a price of 4 euros per kilogram biomass, and then we start to look what could we do to reduce those costs. And what we have to think of is that we did not have to buy the nutrients, but that we could get them for free if you use residual nutrients. That we should not buy the CO2, but that we could use, for example, CO2 from flue gas. We used in our estimations the productivities in photobioreactors like they are now published, and it were all not published but reached in practice. Rather conservative estimations, and we think we can improve that a little bit, and we added that to our calculations. And the final thing that we added to that is that we moved the plant to a more sunny place than the Netherlands, and we even passed Spain in that respect. But we kept it in the Netherlands because we have some in the Caribbean. We have some islands, and there's an island of Bonaire. So we took the solar conditions of Bonaire. And if we did that, we could reach a cost price of production of about 40 euro cents per kilogram of biomass, which is very competitive, which is not possible today. But on the other hand, it is also not rocket science. It is a combination of a number of things that is in principle possible. So with the development time of, let's say, 5 years, we would be able to reach that. Well, is that interesting enough for biodiesel as such? We don't think so. It is still too expensive for biodiesel production. And for that reason, our message is very often in most of our projects that we have to make use of all the value that is present in the biomass. And a part of that is lipids that you can use for biodiesel, but the other components you have to use as well. If you would not do that, you would even cause an environmental problem. What to do with all the proteins if you have to throw them away? You have to do something with that. So what I assumed, and this is just a very general composition of biomass, nothing optimized at all, but just more or less a random choice. Biomass consisting out of 40% of lipids, 50% of proteins, and 10% of polysaccharides. And the calculations here is just very broad. I mean, you can do that more detailed, and you can go into detail into different products as well. The lipids you can, of course, use for biodiesel production. On the other hand, lipids are for some processes also interesting for the chemical industry. What we use, we collaborate, for example, with AkzoNobel in the Netherlands, and we have a project on the production of lipids in microalgae to be applied in coatings. Well, that has a much higher value than lipids for biodiesel. So we looked in this case to the combination of lipids to be used for biodiesel and for coatings. Proteins, the same thing. You can use the crude protein and do something with it, but you can also try to fractionate it. And in this case, we fractionated it in a water-soluble fraction, which has food characteristics, and a water-insoluble fraction, which has feed characteristics. And they both have a different value. Polysaccharides, hopefully we do not produce that much of it if we produce biodiesel, but has also a certain value. Microalgae production, you can also combine with wastewater treatments. And normally we pay for our wastewater treatment, we pay about 2 euros per kilogram of nitrogen removed. And then it's even cheap. I think in most cases it's more than 2 euros. If you produce 1,000 kilograms of biomass, we remove 70 kilograms of nitrogen, so that gives a profit. The oxygen produced at the high concentrations also has a certain value. Well, if we add up all those values, we get, in this case, a calculation, a total value of the biomass of €1.65. And the cost of production was estimated to become 40 euro cents. So you see there is room between that 1 euro and 65 cents and 40 euro cents. Still you're not there because you have to do the extraction, you have to do the refining, which adds costs to your process as well. But at least this gives the room to see that if you apply biorefinery, biodiesel could be one of the products, and it may end into an economically attractive process. Well, these two slides is the basis for our research program. It determines that we have to work on biorefinery. We know the bottlenecks in the systems, and based on that, we designed a research portfolio. And these are all the topics where we work in, and I will not cover them all today. I will just, the ones that are highlighted in yellow, I will say a few things about. But things that we work on is photobioreactor design. We have to produce the biomass efficiently. We look to oxygen removal and CO2 supply. The main reason for that is that oxygen can be inhibiting for the process, and on top of that, the addition of the removal of oxygen or the addition of CO2 costs you energy. It's an energy-intensive process. If we want to apply it in wastewater treatment systems, we are interested in biofilms. Control of primary metabolism, I come back to that. Harvesting and oil extraction. Biorefinery, well, the reason that we do it, I explained. Design scenarios and algae park. And algae park is not yellow, but I will nevertheless I will say a few things about that. Photobioreactor design. Our group works mainly on closed photobioreactors. The reason for that is that we think in the end they can be cheaper, the productivity is higher, and your water management in a photobioreactor is better controllable than in an open pond. Nevertheless, other groups work also with open ponds, and I do not want to add anything to the battle that was mentioned between open and closed. I'm just saying that we are working with closed systems. A lot of our research is focused on the maximization of the photosynthetic efficiency, or in other words, we would like to produce as much as biomass per surface area as possible. And if we look to effects like high light intensity, for example in Spain, shading, Emilio showed the tubes that were on top of each other, so you have some shading effects. We would like to work at high biomass densities to reduce the harvesting costs. We put a lot of attention to energy input, because at the end we would like to have a positive energy balance. Growth inhibition, light guides. Light guides is to bring in light deeper in the reactor. Flashing light effect, I will not explain that, it takes too much time. And variations in light intensity, because not every day is the same, and even during the day the light intensity varies a lot. What is the real productivity? The maximum photosynthetic efficiency that you can reach, well, in photosynthesis is 9%. 9% of the energy content of solar energy can be stored into chemical energy. That's the theoretical maximum. In practice, we never reach that due to all kinds of reasons. For example, you have a part that is lost simply because of reflection, because of the angle that the sun shines on your, with the angle that the sun shines on your surface, you lose a part. In the night, the opposite process is taking place, so you lose a little bit over there as well. And the biomass itself also has their maintenance requirements to survive. So in practice, what we think we can reach theoretically is something like 7.4% photosynthetic efficiency, and which is still a lot because we are far away from that. What in practice? In a closed photobioreactor, 3 to 4%. So what is our task? To realize that we get from that 3 to 4% to 7.4%. That you have all kinds of engineering principles to try to realize that. But that is our playing field. How do we do that? Well, we do that, for example, in collaboration with the University of Welba. And Maria Quaresma, who is also here in the audience, looks at the production, or the possible production of microalgae at the solar conditions that they have in Welba, and she looks to a factor like dilution of light. I will come back to that in a minute. By vertical flat panel reactors, as an example, she imitates the day-night cycle, so she follows the history of the day in a cultivation, and we use in this case a model system, chlorella, simply because it grows quickly. Well, this is the reactor type that we use, and we mimic the situation outdoors via LED illumination. Well, what has Maria done? She mimicked a day in June. So part of the day, oops, back was this, yes. Part of the day it is dark, sun comes up, at noon the light intensity is the highest, and then it goes down. Well, if you grow algae at the light intensity that falls on a horizontal surface area in Welba, you get a photosynthetic efficiency of 4.2%. Pressed the wrong button. Of 4.2% in average. And remember that I said, well, 3 to 4% we can reach in average, 7.4% in practice. So 4.2% at the high light intensities in Welba. If you put the reactor vertically, then the sun comes in at a certain angle, and you dilute in that way in the light intensity. The light intensity on the horizontal surface is then maximum something like 1700 micromoles per square meter per second. And if you put it vertically, you reduce the light intensity at the surface of your reactor. And then this is the light history of that reactor that is placed vertically. And the average photosynthetic efficiency that she got on that day was 6.5%. So by diluting the light, just by putting straight up your reactor, you reach a higher photosynthetic efficiency. And this is a mechanism to bridge that gap between the 3% that we can reach in practice and the 7.4% that we hope to reach in practice. All to make your productivity in your bioreactor better. Of course, this is a lab situation, and the reactor is steel and glass and sterilizable, so it's terribly expensive. So how to translate that to practice? And there are two examples of companies who do that. And I give you an example in the United States, Solix Biofuels, and one in Belgium, Proviron. And what they do, what they have, is they have systems with flat panels. These are also flat panels. They are submerged in water. And because they are submerged in water, you can work with very thin materials so that the investment costs in the end of those systems is low. They are submerged in a large volume of water so that you have a kind of temperature buffering capacity so that you do not have to control your temperature during the day. It levels out over the night and the day. And in these systems, the principle that Maria developed in the laboratory, you can implement in practice. Well, two of those companies are doing that at this moment. Then you have produced your biomass, but what we are very much interested in is the product that we produce in the biomass. So what we are at this moment are doing is developing metabolic models to design a process to produce certain components in the right way. It has been indicated today, for example, that lipid production, you can produce lipids towards high concentrations, but the productivity decreases because lipid accumulation and growth are not coupled. We try to understand that. And you can try, these are complex things, metabolic models or the metabolism in cells. So what we are doing is we model the whole metabolism in microalgae in order to understand what is happening, with the objective to design your process, or maybe even to design your algae to accumulate lipids, if you want to produce lipids, or proteins, or colorants, depending on the application. Well, this is so on the one hand we develop those models, and on the other hand we have well-controlled photobioreactors in which we can make nice balances over your system to really check whether what has been calculated is also the case, is also what's happening in practice. We do that initially with a model organism, Chlamydomonas reinhartii, because the genome is sequenced, the relation between the genome information and the pathways are known, and we built such a metabolic model. That's the phase that we are in now. That model has been built, it has been tested, and it shows at this moment the results that we expect. So in practice, we can get the results that we predict from the model, we can get in photobioreactors. So that is the basis of the system. We understand, or we start to understand, how the metabolism works, and now we would like to steer the metabolism in a certain direction. We also would not only like to do that in Glaminomonas, but also in algae that are more interesting for certain applications. So what we are doing at this moment is to develop, well, that genome-based metabolic flux model for Glaminomonas has been developed. We are starting to work now on metabolic flux models and lipid accumulation in green algae. In diatoms, we are doing that as well. And for another project, we are doing that in colorants. And after summer, we will start on Botryococcus. Yes. Final topic of the research, and then I come to Algae Park, are design scenarios. So we looked at the reactor scale to produce as much as biomass as possible. We looked at the quality of the biomass. We would like to control what kind of biomass is produced. And now we look at the whole system, the whole logistics of the system. Producing microalgae for diesel production and transporting the CO2 from Rotterdam to Bonaire, for example, is not the case to go. I mean, you have to bring all those components together. Water, light, CO2, nutrients. It's going to be large. You need large surface areas. So how is that going to be? So what we are at this moment doing is developing scenarios for design for production of energy carriers at a very large scale. And why? Well, the logistics, the complexity and energy use of supply of materials is tremendous. And research issues in this case are sustainability. Well, I heard today already a few times life cycle analysis. I really think it is important that we dig into that. So that if we do not develop a sustainable system, then we did not reach our goal. Scale and location. So we're not saying that the Netherlands is the ideal location. We're not saying that Bonaire is the ideal location. It's not only about light, it is also about supply of other resources. Algae Park. Recently a project has been produced called Algae Park, and that's Algae Production and Research Center. What's the objective of Algae Park? There are so many technologies, and every laboratory has their own bioreactor system, and everybody claims that their system is the best. But we do not really know. They never have been compared at the same scale, under the same conditions with each other. We would like to develop a competitive technology. Traditional photobioreactors are too expensive to produce biodiesel. We would like to develop cheaper processes, and not only in the laboratory, but also to go outdoors and to do it at a larger scale. And for that reason, we started this project. And it's to build up the more applied part of a photobioreactor design, to translate research in applications and to acquire information for full-scale plants, to have the total information of that. In the end, we would like to develop, of course, a competitive technology, cradle to cradle, to be applied inside but also outside the Netherlands, and for that we have that research program. I will show a couple of things of that. What we have at this moment is this. We do a lot of fundamental research. At this moment we have a large collaboration in this program with Wijffels. 14 companies participate in that. And we would like to go in the end to demonstration plants. Well, I'm from university, and we would not like to go to this scale, but industry has to go to that scale. And we need an intermediate phase. And for that, Algae Park works at 2.5 square meters and 25 square meters. And the idea of it is to develop the whole process chain. So not focus only on production. To develop experience with systems. To have information for design of full-scale plants, like I already mentioned. We also would like to compare systems. Open ponds with horizontal tubes, with tubes that are piled on top of each other with flat panel reactors, for example. Compare strains, comparing feeds, and supply of biomass for further processing, which is also often a limit, and further processing, and in that case, we especially think about biorefinery. Main features of Algae Park. We will install at a large scale 4 different systems that can run in parallel, so they completely will be operated in the same way. The fundamental aspects for successful operation and scale-up of photobioreactors to commercial plants. So we want to gain knowledge to do that, do all the analysis, etc. And flexibility, because during the time we would like to develop those systems. We start now with 4 systems. We hope to add systems to that, and systems that do not function in the end we would like to replace by better systems. The budget and time plan, the facilities will be built this year. It is a 2.8 million facility. The first test runs will even be done this year still, and then there is a research program in which 14 companies participate. And what we aim at with this facility is to be a kind of test center for microalgae technology. That is what the Dutch government and the companies set as an objective. The conclusions of my talk. What we are trying to do in Wageningen is to develop new technology for cost-effective production of fuels, but not only of fuels in combination with food, chemicals from algae. In our opinion, it requires a multidisciplinary approach. You should not only focus on the photobioreactor, not only on the molecular biology, not only on the biorefinery, but on everything. We are active in all these disciplines, or we collaborate with groups that are active in those disciplines, and I showed you 4 of those examples. Also, in the Netherlands, microalgae is a hype. And you cannot read this. It is in Dutch. It is in a citation from a newspaper from the province in Groningen. And that's one of the starters in this field. And he starts the article with that algae are so promising and for biofuel, and this is going to be the way. And he started to do tests, and he did 4 months of tests, and he discovered that it was pretty difficult to grow them. It was pretty difficult to extract the lipids, and it was pretty difficult to convert the lipids into biodiesel, and concluded that after 4 months of experimentation, microalgae is absolutely not the route to go for biodiesel production. Having said that, the timelines that we think of to apply it at commercial scale for diesel production are in the order of magnitude of 10 to 15 years and not 4 months. Thank you very much.
Thank you very much, Professor Wijffels, and also we hope your new facilities will be very soon in operation and a reference for everybody. So let's start. Vamos a poner también un poco de tiempo, 2 o 3 cuestiones. Profesor.
René. Muchas gracias, René, por esta magnífica exposición. Me gustaría preguntarte, el proyecto este de Algapar es un proyecto con fondos públicos, he entendido 6 millones de euros, 2 millones ahora y 4 millones para investigación. ¿Me puedes explicar el proyecto? Porque luego también he visto que hay de compañías. Es un centro de investigación y de demostración público, privado. Parece que eso es público, independiente, y que ahí pueden entrar las compañías que quieran a experimentar lo que ellas consideren oportuno para dentro de la biotecnología aplicada. ¿Es correcto?
Yeah, that's correct.
Pero los 6 millones de euros son del gobierno holandés.
Shall I explain it?
Muchas gracias.
Yeah.
Eso es lo que quería oír.
Yeah, the investments are it are 2 projects. One is the research, and one is the investment. The investment is it's fully covered by the Dutch government. Well, partly by the Ministry of Agriculture and partly by the province of Gelderland, where Wageningen is located, and partly by Wageningen University itself. And the research program is 50% covered by the Dutch government, and 50% covered by industry. It is an open center. That does not mean that everything is right on the street. But the companies, they pay a fee. They have to pay a fee every year, and they have to commit themselves for a period of 5 years. And they can enter as time proceeds. That's how many Dutch projects work. And you start with 14. Some of the companies will want to leave us, and others would like to join. And that is in principle possible. That is also not restricted to Dutch companies. I mean, the reason that the Netherlands invests in these kind of initiatives is that to develop competitive technology. And if you need foreign companies in that, foreign companies can be allowed. And in this case, it is also the case. I think that 50% of the companies is not Dutch, and some of them are not even European. Can everybody enter? Well, yes and no. I mean, you have to pay the fee, and besides that, it is also a facility where one-to-one contract research can take place. The owner of the facility is Wageningen University and Research Center, and the objective is to expand, so to attract new companies to enter, and even start-up companies. SMEs get a lower entrance fee than large companies, and that kind of things.
And your commitment for this year is to set up 4 different technologies running and this year?
4 different technologies running this year, tested at 25 square meters. It's not that large. And we also have a baby room of 4 systems of 2.5 square meters.
Thank you very much.
¿Alguna pregunta más? ¿Algún deseo irrefrenable de una última pregunta? No? I have only one clarification. When you mentioned the added value of 1.75 euro per kilo, how do you contemplate in your calculations of external added values the CO2 sequestration that you may have if you connect your process to because CO2 sequestration is going to be a big business in the future, so and this has a value as well. So
yes.
Is this contemplated in these calculations?
It is contemplated in the calculations, but you don't see it because if you have a CO2 tax of 25 to 75 euros per ton, it almost does not add to a cost reduction.
So it's peanuts.
It's peanuts, yes.
Okay.
Yes. Or you have to increase the taxes.
No.